Okay, let's get started. Sorry, we had to cut lunch down by about 10 minutes or so, but hopefully you've managed to get something. So great pleasure to introduce Tony Wood. Tony and I worked maybe 10, 15 years ago on a particular issue, and then Tony was also involved in the British Pharmacological Society when I was there as the president-elect, I think. So Tony is a medicinal chemist by background, but now tells me a technologist. And Tony has got many accolades, including being Fellow of the Royal Society of Chemistry, Fellow of the Academy of Medical Sciences, and various other bodies. And Tony is now the Chief Scientific Officer for GSK. And Tony is going to tell us about how we're going to create a better, healthier future. Thank you, Tony.
Thank you, Sir Munir. So if people want to grab their sandwiches and eat while I'm talking, please do. This should take about 25 minutes, and then I'm looking forward to a question and answer session, although I do note that I didn't get to have any friends with me to answer the questions. Before I start, just a reflection on this morning and what I'm going to do. I'm going to talk at quite a high level, and what I want to do is provide for you, if you like, the view of a technologist and a drug hunter. That's how I think about myself, and what those opportunities that we've been talking about today and broader ones in data might bring to a different approach to how we can think about using science and technology and collaboration to solve some of the problems that lie ahead of us. But there are a few things that Janet picked up on this morning that I want to reflect on, that I grew up, as Munir said, as a medicinal chemist. And you flatter us, I think it was more like 25 years ago we were doing that work together. And at that point in time, which was the sort of late 80s through to the beginning of the 2000s, we were very much in the heyday of the small molecule population health era of the pharmaceutical industry. And of course, what that did was left us with a set of systems and processes and devices that were very much constructed during that period of time. And I think if you're picking up on one theme from this morning, it's a realization that those systems and processes really aren't fit for purpose today. I mean, let me give you an example from the standpoint of some of the technologies I'm going to be talking about. I'm not going to get into a lot of detail. We can do that if you want to get nerdy in the Q&A. I'm very happy to do that. But when I started, the single unifying intervention was the small molecule. Largely speaking, we couldn't measure disease. We got only close to understanding the pathobiology of disease when we managed to successfully intervene in it. And as you all know, the number of times we actually do that, we're limited. Fast forward 33 years, which is how long I've been at this now, and we find ourselves in a position where our ability to measure and intervene with disease is just unimaginable for me. And I think what that brings for us is an opportunity to think about re-engineering the whole process. It no longer being that horrible step chart diagram that you usually see from people like me, and I can promise you I'm not going to share, in terms of how you start with target validation and end up in a phase 3 study that, if you were lucky, you guessed all of the things that our friends in regulation and health tech are the questions that are going to be important to them. That world really has to change for us now, because we're entering a position whereby we can do something we couldn't in the past, and that is measure the disease before we seek to intervene. And we can measure that disease at a range of different scales and at a range of different details. Now, there are massive technological, structural, we talked about them this morning, problems that lie in front of us to bring all of that together. But I'm going to use the next 20 minutes to not worry about all of those, if you don't mind. That is my day-to-day life, and it's delightful to be here for a day and not have to worry about that, but rather spend a little bit of time imagining what the future might be. And I'll bring that to light in terms of the approach that we're taking in R&D in GSK. So what I'm going to do then is imagine about how we might transform, and I'm going to talk about research, but you'll see there are echoes into development. You'll forgive me if I don't go into the aspects of access and regulatory data and science that we were talking about this morning. You know, for me, research and development has always been the heartbeat of what we do in the industry. For me, it's where curiosity and creativity meet to generate hypotheses that we have the privilege to be able to test, and some of which end with tangible solutions that help patients. And as I've mentioned, today more than ever before, R&D is being transformed, supercharged, if you like, by incredible advances in science and technology. Imagine if we can go from the world of 33 years ago when I started, when it was all about small molecules and Jim Black-based translational pharmacology. Trevor, you will remember that very well. Not to in any way diminish the achievements of that era, but the challenges were very different. So now we're in a world where I can measure the transcript of a single cell in a disease from a patient and know pretty much the phenotype of that cell as a consequence of it. If I could link that through the outcomes that our friends in HTA and others are looking for, there is the real opportunity. For us, it's all about putting the patient, though, at the center of that. And I'm going to come on more to that. If you put the patient at the center and recognize that we don't follow that old step chart, but rather seek to measure and then intervene, it drives a necessity for partnerships and collaborations that you'll see I'm trying to build, but I think are critically important to future success, and what makes me so excited about spending the day with you all today. So, you know, for me, it's about imagining that future. This is part of my job. It's the bit that keeps me sane, quite frankly, dealing with the possibility, trillions of data points at our fingertips to unravel the mysteries of biology. Health data already represents something like 37% of the entire data on the planet. That is only going to go up exponentially. And so we have a wonderful opportunity. Imagine then if we could pinpoint where diseases start so we can stop them in their tracks. Are the Mendelian rare diseases that we've just been talking about the hints of the underlying powerful pathobiology that could present an opportunity there? Imagine with that in mind, and the biomarkers, be they based on molecular targets or more sophisticated measurements of underlying pathobiology, that through that we could pick the right medicine for the right patient at the right time. That's the future that I'm spending my time thinking about. I don't think it's a distant dream. We went from small molecules to this in 30 years. We have all of this in front of us now. And really, for me, it's about if we can put the patient at the center of what we do, we can solve more of these problems. So, with that in mind, and seeing if I can work the slides, which I'm delighted I can, where do we start? Well, I think, as I've been illustrating, the first start is I think we need fundamentally new approaches to drug discovery and indeed development. We can talk about the aspects of data and development later if you like as well. And look, there's no doubt that we've made incredible strides in health during the period of time that I've been involved with the industry. Life-changing medicines and vaccines resulting in people living longer than before. Those trends are only starting now to change in the wrong direction in certain places. You know, I don't know how many of you followed age demographics, but there are already more 60-year-olds on the planet than there are 5-year-olds. Unfortunately, though, and for me this is a challenge that, if you like, motivates me to have the things that I'm talking about be my constant thought partner. My wife complains that it's too often that they're my constant thought partner. Are that despite that, rates of chronic disease, obesity, disability are rising. You heard this morning that we are able now to characterize rare disease with a degree of precision that is fantastic, but it's just simply not good enough that we're unable to do anything about it. I work very closely with our colleagues in ViiV for HIV, and their approach is no patient left behind. And I think we need to learn how to work across that spectrum for that to be something that we all sign up to. I spend a lot of time over Christmas working with my colleagues and friends in the UK healthcare system as part of pulling together a task and finish group for health and R&D. And what became increasingly apparent to me as we worked through that are some of the realities and inequalities of an aging population. You know, more than half of adults aged 60 and over have multi-morbid conditions. You know, as a drug discoverer, we kind of start with, here is the disease, but it's conveniently forgotten that that disease rarely shows up as just that disease. The other thing that I find particularly frustrating in that, and it speaks to my background, I grew up in the mining communities of Newcastle and Gateshead. I'm the product of a free education in the UK. I'm back here because of that. Spent 7 years in Boston, teaching a little bit at Harvard, working with friends there, but I'm back here because I want to change this dreadful inequality. We're getting older. Lifespans are reaching early 80s. The truth of the matter, though, is you can separate somebody by literally 50 miles on a motorway in the UK. You can go from an affluent community where you will be healthy for most of that 80 years to a deprived one where you will be ill with a multi-morbid proposition pretty much from the age of 50 onwards. So, of course, that's something that none of us ought to be comfortable or relaxed about. And I think it's critical that we work together to solve that problem. I find inspiration in the multimorbidity from a hypothesis that says it doesn't arise just because we have issues of lifestyle environment to deal with, but that fundamentally the diversity, the ontology, Greg, you probably come across this all of the time, the ontology of disease. The last time I looked at DSM, I think there were 17,000 different conditions described. More conditions necessarily than there probably are active genes. Of course, that can't be right. One thesis for multimorbidity then, I think, is that underlying pathobiology cuts across a lot of these diseases and provides opportunities. So people and economies are depending on us for solutions, not just to live longer, but healthier lives with medicines and vaccines that give precious time to enjoy the things and be with the people that we love. I lost my father, the man I knew, when he was 55 years old to a heart attack. He lived on for 20 years as a shell of the person he was before, reminding me every day of that consequence. That's why when I get up in the morning, I'm thinking about people like him. I'm thinking about the cancer patients who I meet. One particularly in mind, I can remember him standing at a lectern saying, he knows it's difficult. It's difficult for all of us, right? You know, but every day that guy gets up, he deals with cancer. So, and I like to remind myself that cancer and all of these other diseases don't take the weekend off. So we've got to lean into this. R&D, of course, is an inherently challenging business, and success rates for drug development in our industry remain stubbornly low. A lot of what we're talking about, Janet, you pointed it out in terms of harmonization. These are not new problems. They're problems that are stubborn ones. But I do think technology offers us an opportunity for the first time to perhaps break some of those stalemates. Of course, it also takes an unacceptably long period of time, 10 to 15 years if you're doing it well, to go from an idea to a medicine. And I'm lucky enough to have discovered a medicine at the short end of that spectrum for HIV that had a number of features that I think we can echo into what we want to do for today. So the question is, and sorry, excuse me, this is a problem with speaking after lunch. How do we break that cycle of inefficient, unpredictable, and traditional ways of working to operate with more pace, more precision, and ultimately, of course, a greater probability of success? And I think this held in an approach that offers not the possibility of not just treating symptoms of disease, but intervening earlier to alter course, perhaps even preempt disease. One of the promises, and we're going to have lots of fun about things I say that you disagree with in the questions and answers, I hope. One of the promises of this perhaps underpinning common pathobiology is that there are areas where one might imagine defining that pathobiology when clinical symptoms become apparent, but subclinical expression of that biology is apparent for adult life, and could we get ahead of disease by treating that? We have a series of medicines that target IL-5 and deal with hypereosinophilic conditions. Hypereosinophilia is a sign that could become a treatable trait for the future. Having established the context of benefit-risk in patient populations where it matters and where burden is a significant issue, for example. We particularly focused recently in our COPD studies on the burden of hospitalization associated with that disease. For those of you who don't follow COPD, it is a pretty horrible disease. If you're admitted to hospital, you've got a 10% chance of never leaving. Your 5-year survival rate is 50%. So imagine if we could get ahead of a disease like that to prevent exacerbations occurring at all. So there are the problems. I'm now going to have a little bit of fun because I'm a passionate cyclist as well as a technologist and a drug hunter. So what we're going to do is talk a little bit about some of the parallels between the two. I promised Simania and the organizers that I wouldn't get into technical nerdiness and I'd keep this very high level. So if it's frustrating you, bear with me. I will answer questions at more detail, I promise. So many of the characteristics that drive me as a scientist also drive me as a cyclist. Both fields require commitment, discipline, focus, resilience, and a passion for pushing yourself to the limit. I believe there's a lot we can learn from this world of cycling, and it's back to the three things that I've been stressing and will continue to stress through the presentation. That is the revolution that we're seeing in science and in technology, and at the end of all of that, the essential nature of collaboration if we're going to bring those two together in a way that will benefit patients. So let's start with science. I don't think I need to convince anyone in this room about the immense and disruptive power of science. And throughout history, breakthroughs have transformed our understanding of disease and our ability to approach that. I describe myself as a technologist, not a medicinal chemist, because people look at medicinal chemistry as a subject. It was just the first technology that had a significant impact on drug discovery. Throughout history, you see examples of that. This one amuses me. There's an American cyclist called Charles Miller Minute Murphy. And he became the first person to ride a bicycle for 1 mile in under 1 minute. Just do the arithmetic on average speed for that one while I'm describing it. Today, over a century later, a professional cyclist manages around 2 miles without the particular environmental assistance that Charles used. You can see it in the diagram. I'll tell you what it was. His mile was made possible because his analysis was anchored in science. And Charles had the wisdom to realize that it was all about wind resistance. And that he figured correctly that if he rode in the strip stream of the Long Island Railroad train, having been up and down on that railway line between Boston and Connecticut, between Boston and New York and Long Island, it was obviously a lot flatter in those days than it is now. But he understood that removing the wind resistance and bringing vacuum would allow him to solve that problem. So, what's the equivalent of that I see right now that presents an opportunity for us as far as drug discovery is concerned? And we've been getting at it this morning. It's this opportunity now that we can have a deep understanding of the underlying drivers of the condition, in this case disease, that will help us disrupt the status quo and make breakthroughs possible now that on the surface might seem impossible. I'm passionate about the relationship between Mendelian phenotypes and what they offer, particularly in neurodegeneration, where it's very, very clear potentially to solutions in more common conditions. Now, easily said, difficult to do. Lots of challenges in that, and Matthew and I have spent a lot of time talking about how oligonucleotides might impact on that particular arena. But you can extend the analogy elsewhere. And because of that, then, it will probably surprise none of you that the key components of how I'm looking at the R&D strategy within GSK have some of these ideas at their heart. Advances in human genetics and functional genomics are transforming how we think about the underlying mechanisms of disease, the role of biological processes, common ones like inflammation and senescence, and their contribution to this concept of aging. The role in particular of cell types in driving phenotypes. I was lucky enough to persuade Sarah Teichmann to come and join us for some of her time at GSK. Sarah, and I love this, by the way, Sarah is one of the two leading ladies in the world who have driven the field of single-cell science, her and Aviv Regev. And what comes from that, particularly in autoimmune disease, is the realization that if you can get to the specifics of disease-causing phenotypes and find a way to successfully delete them, then you'll be able to address autoimmune disease in a different way. The era of biologics, for some reason this was on my mind as I was coming down here in the car this morning. We go from the 1990s, the first big single mechanism, anti-TNF. It works, but boy, is that a blunt instrument. PD-1s. It works, but boy, is that a blunt instrument. It's introduced all sorts of regulatory concerns over whether PFS is something that can be relied on versus OS. And now we're in MRD in one of the classic areas where cell type deletion is probably the solution, myeloma. So I think there's just a world of opportunity as we begin to borrow Chris Buckley, who's at the Kennedy Institute in Oxford's way of describing it, the periodic table of disease, which has cells at the origin. That, I think, gives us a fantastic opportunity. And, of course, sitting around all of that, the role of the immune system, either in addressing or contributing to this dynamic. I'm lucky enough to have both vaccines and therapeutics under my purview, as it were. And that brings some very interesting questions. I'm going to mention a study that we're running in the UK later that I think brings that to life. So we're building up an increasing wealth of diverse data, genetics, genomics, gene expression, increasingly nowadays epigenetic regulation and the understanding that that plays in switching phenotypes with age, imaging. I've had conversations already with people here. It never ceases to amaze me that we can get at the molecular origins of biological processes from an image. So all of this contributing to that dynamic. Of course, there are substantial problems that remain to be solved in that. We've talked a little bit this morning about data access. And this is one of the areas where we have got to find a new approach. We cannot use the instruments that controlled interest, intellectual property, access, things like that, as a vehicle for managing database collaborations today. We need a completely different framework if we're going to make that successful. And we do need to be able to share data across all of the groups of interest that we have. So I spend a lot of my day dealing with those prosaic problems that are frustrating but necessary to solve if we're going to, and where I always put myself in that particular conversation, by the way, is the point that was made by one of our speakers this morning. Those people who give their permission for us to use their data in trying to help find solutions. I can see you now. They don't like the idea that we're spending weeks, months, years with lawyers trying to figure out how we do the thing that they just asked us to do. So it's really important that we focus on solving these problems so that we can have scaled solutions. Now, of course, what sits behind a lot of that for us is the opportunity that is presented by, I was deliberate not to get to mention AI/ML until at least I got to slide 5. Because, of course, AIML is a wonderfully powerful technique, and it really does, across these types of data sets, give us an opportunity to interrogate and look for signals that quite simply the human brain could not, given however much time we liked, decipher. The image point that I made earlier is really a classic component of that, but I think it offers fantastic opportunity for us to really completely rewire not only the target identification component, inspired by a much, much better choice of who my patient is. One of the biggest challenges I face when I'm about to sign £500 million away on a phase 3 study are all of the things we were talking about this morning. Who are the people that I'm trying to benefit? How confident can I be in a heterogeneous chronic disease that for all of those people, I'm going to provide a benefit proposition that you'd like? How do I balance that in the reality of a statistical testing hierarchy that Janet and June and the statisticians that I encounter in regulatory discussions are quite rightly going to expect me to demonstrate to avoid the multiple testing problem? All of these things are real challenges that I think only data can help us solve in the future. The Greg's comment on £350,000 or dollars per patient in oncology studies is pretty on the mark. And I would like to be able to do that. You know, our endometrial cancer medicine that hit the news from a collaboration with Memorial Sloan Kettering 24 months ago in rectal cancer cures people with dMMR rectal cancer. One of the analysts who I see more frequently than I would like, it's a part of my job that I love them all, honestly, she is a young mother who was cured by Jemperli. So when we get it right, it clears a lot of the debates because benefit risk is obvious and it's quick. To your point, you don't need thousands of people and a statistical analysis. So how do we solve the problems to get to this? Now, for me, where am I pushing with this? It's this back to this idea that technology now affords us a very different way of thinking about how we go about defining the right targets, finding the right patients, and then ultimately designing the right intervention for those patients. And that is that we need to start thinking about translational medicine in a different way. And I started using this term without really thinking about it, to my regret, and got involved in a lot of conversations, and people would say, wait, but we already do this, Tony. So do we really? What they really mean is the old world of getting a medicine, taking it through a phase 1. That's not what I'm talking about. What I'm talking about now is the opportunity, if we can put the patient at the center of what we do, and I couldn't agree more, Janet, with what you said about we need to spend more money doing that. That's really the issue here. Then we can start with translation in the right place. We iterate from the clinic backwards, not the lab forwards. And again, easy to say, difficult to do. We've got an enormous amount of work to do to bring, quite frankly, the single best informed expert about what we're trying to do into the conversation. The patients really are the people we should be listening to more. I would love to make more of PROs. And we only get that information by talking to the right people. So bringing patients to the center of what we do. Some of them, because of tragic and very understandable reasons, are willing to go even beyond that, to regularly visit clinics, to be biopsied, to be measured. And that is at the heart of, I think, how we solve this problem. Now, of course, common disease has an inconvenient feature associated with it that I can't simply select 100 people at random and biopsy them in detail and I'll just produce a lot of mess. That this was the conversation that we were having this morning, that with increased ability to measure and characterize did not necessarily come increased precision. So how do we deal with a subdivision of heterotypes that are inspired from real-world evidence to allow us to start to make the right decisions in that area. You could look at, and there are a variety of different ways of approaching this problem, but one could look at diseases whereby we characterize endpoints through multidimensional devices. Neurodegenerative diseases carry all of those. You can look at the distribution of data into those domains, and it gives you hints as to the underlying pathobiology of the disease. Doesn't work all. I would love to be able to do it for lupus. Lupus is just too complex. If the word protein appears in the description of your disease, you pretty much know you're in a difficult position. But look, all of this, I think, is possible, particularly as we recognize that there is a revolution occurring in how we not only measure disease in a secondary or tertiary setting, but increasingly from the home to the high street to primary care through wearables, or to bring on one of the UK's particular strengths through our ability for Ragib and the guys at Our Future Health to begin to engage in a population more broadly. I'm going into all of this because I want to try and describe for you that when I go to work, I don't worry about the 13,000 people that report to me. Of course I do. But I can only do what I need to do if I can engage the entire ecosystem of people represented in this room. The UK has a unique, perhaps not unique, but I think a short-lived opportunity to begin to make a big change here. We are big enough to have the diversity that we need, and small enough, perhaps, to integrate it all. We have world-leading academia. You'll see on some of the collaborations that I highlight, as well as other friends around the rest of the world, that brings together this union of academic medicine, physical sciences, and data sciences to bring a wonderful opportunity for new insights. Having said that, we need to focus if we're going to do that. So I'm starting to progress this vision at GSK. A few examples. Duncan's in the room. We have a collaboration at Cambridge that we call the GSK Translational Immunology Collaboration. What you're going to take from this is none of these collaborations have memorable names, but they all have catchy acronyms. CGTIC. It's a 5-year research partnership, and what we're looking at there is really kidney and respiratory disease, but I'm at the underlying pathobiology of fibroinflammation, not the old world of collagen deposition. We've been there, it doesn't work. Fibrosis is the second best response to an unresolved inflammatory condition. What I'm keen to understand is how we guide wound healing in inflammatory conditions into a healthy response. And increasingly, the single-cell proposition associated with that is becoming apparent. At Oxford, something that Richard Cornell and I set up called the Oxford Institute for Computational and Molecular Medicine. I told you, didn't I? They don't have catchy names. IMCM. And what we're doing there is putting money into the identification of new clinical instruments for neuroinflammation. And I think this for me is really important. The traditional view of people in my job was that goes on in academia. I'll invest when I'm thinking about making a medicine. And of course, it introduces all sorts of tensions when you bring both the biomarker and the medicine together at the same time. What we need to do is work together to solve that as part of measuring the patient proposition, and we're investing in doing exactly that in neuroinflammation. So our goal then for all of these is to deepen our understanding of the disease and its progression. When we get it right, that will bring major benefits. It'll drive insight-driven discovery and development, hopefully to resolve some of the issues we were talking about in terms of the size of studies. It's why I think orphan diseases are just such a wonderful model. You get a very tightly described population, and you get an endpoint that you can measure quickly. Well, why can't we do that by the subdivision of common heterogeneous disease as well? Isn't that a better way to go forward? From all of that comes acceleration, and I haven't yet mentioned the aspects. Of course, before I go there, sorry, what this does is puts the patient at the center of our research engine. Not the laboratory, not reading Nature, but rather patients and the data that come from it, and collaborations that are different. I'm going to mention, I want to just quickly highlight tech, though, in all of this, because what we haven't talked about at all is the opportunity for intervention once we've decided on that target. Here, the generative AI revolution is only just beginning. The ability to do what used to take 6 years in 6 months for small molecules, programmatic modalities like oligonucleotides, generative design for biologics. These are all with us right here, right now. It's almost now become a commoditized component of what we do. I look forward to the more platform-based approaches that are approved on the basis of that platform with cassette substitution as the opportunity to address disease as well. We're a little way off that yet. But we have, as has already been noted, real examples and regulatory pathways in practice right now. So, like science, then technology is going to really transform what we're doing. And another bike analogy, if I may. So, the bike market is going through challenges. It had massive growth during COVID. Post-COVID, it's struggling, apart from one area. The whole world of e-biking, not just powered bikes for people who want to get into cycling and find the initial proposition hard, but also the whole data collection associated with it. That market has a CAGR over the next decade of 14.6%. For those of you who do jobs like mine, 14.6% would be a fantastic proposition for a CAGR. And of course, it's largely driven by connectivity. You can measure pretty much everything from your power output to your glucose utilization, as well as having a powered bicycle. So what can we learn from that example? Well, I hope, in what I've said already, I've been through what we can learn from that example. Digitizing disease is a problem that will have massive impact, but one that we have to solve together. At GSK, we're embedding that idea really from the beginning to the end of what we do. And that applies, I think about it in these four buckets. I'm not going to talk any more about patient choice and target identification because you'll be bored of it and we can get into more specifics in Q&A. But molecule design, entirely a predictive process now. So the panoply of different approaches that I have to intervene is unsurpassed. There's one area that I would really love more of, and that's conditionally responsive medicines. You pick up some of the classic targets in neurodegeneration like TREM2 that influence microglial function and the polarization that occurs there. I know what I want to do, I just don't know how to do it. And so that, I think, remains as an opportunity for us. But, increasingly, we're using these approaches also to inform clinical trial effectiveness. There are the prosaic aspects of this, recruiting the right patients in the right places with the right characteristics. That's the current focus at the moment. But as I say, if you build on translational networks and collaborations, as I've just described, my vision, hopefully reality of the future, is that we have patients who are with us permanently. By us, I mean the entire community. Translational medicine, in which we're managing their conditions, and then from that straight into clinical studies, set perhaps under the sort of basket context that we were, or bucket context that we were talking about earlier. I think that can only help with the regulatory burden. It can help provide better comparisons for efficacy. And of course, it means that patients get to the right medicines earlier, which is what we're all about. So, a few examples of having talked about what this might all bring. I'm going to, at a very high level, tell you about what I'm actually doing, as well as the collaboration. So, for oligonucleotides, and I'll talk here about steatotic liver disease, which has an enormous burden. 5%. It's a chronic progressive disease that affects 5% of the global population. Has a significant health burden associated with it. At its heart, there are metabolic and alcoholic drivers. MASH has recently been recharacterized. You've all, I'm sure, followed that. Alcoholic liver disease is an enormous problem. 12 million patients globally have MASH. 26 million have alcoholic liver disease. It accounts for nearly half of all liver-related deaths. And those two together are the biggest cause of liver transplantation in the USA. So they've traditionally, of course, been seen as difficult diseases to treat. Now we're seeing some progress in MASH, and all of the way, by the way, all of this is on the background of I acknowledge there is a future called post-GLP-1. But what is very clear, even in MASH, is when you look at the more cirrhotic end of the spectrum, that F4 stage, GLP-1s don't help. Actually, there's argument to suggest that they're deleterious. So we're interested in that stage of the disease. Obviously, we start in F2, F3 in EMASH because that's where the regulatory pathway allows. We have a number of targets in this area that have really strong genetic credentials. They're PCSK9-like, if you want to draw a parallel, and they're oligonucleotides. The great thing about oligonucleotides in liver disease is twofold. One, they tend to go to the liver, and two, I don't need to know any functional biology. I simply need to know, do I want my transcript to go up or go down? Now, obviously, I'm not going to stop there. I'm going to learn about the biology for biomarkers, but this presents wonderful opportunities for us to address a disease which brings enormous burden to healthcare systems. The other one I've mentioned already, COPD, and really this is about understanding the heterotypes of that complex disease. There are those who are responsive, and it's heterogeneous in two dimensions, if you like. They're the underlying pathological drivers. Are you eosinophilic? Is your disease predominantly infection-based and increasingly Th17-driven? Are you simply bronchitic? Are you emphysemic? You're most likely mixed. So all of that needs to be brought together in how we think about the development of treatments for COPD. And the good news is that we have the collaborations that are increasingly allowing us to approach that problem from the phenotypic angle that I mentioned. Cancer, similarly, I'm probably running out of time, so I'm going to hurry up and get through that. Obvious examples there. The world of ADCs is blowing up. B7H3, B7H4. If you haven't followed that, that is a solid tumor opportunity. And I started working in Pfizer and Mylotarg 20 years ago, so ADCs have been with me for a large part of my career. Targeting malignant cells, understanding the tumor environment that determines how they process ADCs and respond to cytotoxins is the next horizon there. So much more to come on that front. And then lastly, I couldn't finish this with just another mention of AI/ML and then quickly collaboration. I talked about AI/ML and integrating data. Hands up who uses an AI/ML-based product to interrogate and read the literature right now. If you don't, you should. Things that used to take me 2 weeks now take me 5 minutes on a Sunday morning. And it's gone from doing a great job of summarizing to now reasoning and suggesting experiments. That's just the beginning. These agents will not only do that, they'll direct experiments to the future. And they're AI colleagues. They're going to get us to a part where we can do more of the exciting stuff that we want to do in our jobs. So for me, it's a truly exciting time to be a scientist. It's exciting to be a cyclist as well. But and then lastly, can't do any of this. We at GSK, we talk about ahead together. And some of these corporate things can become trite, but it really actually means something deep to me, and that's captured on this slide. It's the idea that everything I've been talking about cannot be done in isolation. I've mentioned a few of the partners on this slide. This is, by the way, not the comprehensive list. It is simply a selection of those individuals that we work together with to try and get at the underlying basis of disease, or to challenge how we might approach its resolution in new modalities. That's what our collaboration with my friends at Flagship Pioneering is all about, where I get to sit with Nubar and say, I can't deal with TREM2, get me a situationally specific molecule to address that. So I think I'm coming to the end. Having said all of that then, what's my finish line like? I'm leaning into this, drawing the inspiration from my past experiences with cancer patients, a future where we deeply understand disease. So, and potentially where we don't just treat it, but we preempt it. A future where tech brings new possibilities to get the right medicine to the right patient at the right time. Importantly, though, a future where together we solve these problems. And I think a lot of our, the one thing I have discovered is that no matter who I'm worrying about, who I perhaps might have perceived as being adversarial in our interactions, as soon as you sit down and talk to them and realize that we've all got the same goal in mind, that all goes away. And I think we need to do more of that. So a future, if you like, where we get ahead of disease so that we all live healthier, not just longer lives. That's what I'm aiming for. Thank you.
Thank you very much, Tony, for a very insightful talk. Thank you. So we've got about 5 minutes for questions. Trevor, please.
Trevor.
Tony, thanks for an inspirational presentation. You know, the excitement that you've given us is the opportunity to cure disease and not just treat it. As one of your predecessors at Glaxo Wellcome, when I was at Wellcome, I was honoured to receive 4 pre-Galien, and those drugs are still used, but they're still not cures, they're treatments. But what my question is about the biobanks that exist both in this country and elsewhere. From what you're saying, you'd rather get a known population and create a biobank for them rather than just go to the historical biobank.
Well, I think, Trevor, it's sort of, none of these are monolithic solutions, right? And it's trying to get at the population-based GWAS biobanks tell you about predisposition. I think collectively they enumerate for us the underlying biology and the risk factors that are important for particular tissues. But for the physicians in the room, I don't think any of you would accept that a late-stage, highly compensated disease has got anything to do with what I've learned about genetics. Hence why I want to get into patient phenotyping as well. So it's the marriage of the two that I think gives us the complete position. And increasingly for sequencing, what I'm more interested in is the phenotype-genotype correlation. And the population-based biobanks were good. And look, I'll continue to invest in them. There's a collection of things that I invest in that we've talked about some of them this morning, have a simple characteristic. We all want it, but none of us can afford it individually. Preferably, we all get different benefits from it, and then that's the ideal set of circumstances for collaborative networks.
Is there any organisation that brings together all these biobanks, you know, so that not cost of thousands, but just a few people who coordinate the biobanks?
It's not done. I think everywhere it's happening. You know, I'm hoping we can do more in the UK. Part of the proposal for the R&D thing was that we focus on these translational centres which bring Regib and biobank world of population into, I'm going to call it tertiary, so that we can get into the detailed levels of understanding patient journeys. The US does this well at its individual hospital centres, the likes of Cleveland and Mayo Clinic. Of course, the challenge that the US has is the insurance structure, which means that longitudinal coverage is hard. I think if you're not looking at what's going on in the Arab Emirates, you're missing a point there. There's huge opportunity. Spain is bringing it together. It's all sort of coming together in union. No one has yet solved it, I don't think.
Tony, I love the vision. But can I bring you to a more fundamental concept?
Yes.
As a clinical pharmacologist, I worry about the dose.
Yes.
The dose is either the remedy or poison. And patients I see in my clinic now have got 6 diseases, are on 15 drugs, have impaired renal function, impaired liver function, yet the drug dose was tested in a 40-year-old with 1 disease. Are we overdosing our elderly? How do we change the paradigm?
Well, I think perhaps I might ask the question differently. Have we got the right dose and the flexibility to deliver that dose? I don't think so. And of course, pharmacogenomic considerations as well as drug-drug interactions. We've done our best to deal with that up to now. Again, for me, it's back to having confidence in models. If we take the area you and I have been interested in terms of pharmacogenomics for P450s, there's no reason why we couldn't have a dose which is determined at a pop PK level, but then a set of modifications that we're used to doing in the context of drug-drug interactions, perhaps on the back of a pharmacogenomic phenotype, for example.
Yeah, so that requires a change in the paradigms for industry, but regulators,
And
but
regulators
also clinicians as well.
It's back to the point that you were alluding at. We've got to stop thinking about tomorrow the way that yesterday grew up, basically.
Okay.
Well, Janet Woodcock, first of all, in that point, it's the healthcare system that's not able to deal with this. And that's probably where AI will come in, because individual frontline practitioners are not able to deal with dose modification. They don't have the time, they don't have the background knowledge, it's complicated and so forth. My question is a hard one for you, all right? Perhaps not in your company, but in general. Of course, we know that if we look at this phenotypic and biochemical differences and we subset the patients, we'll get more effective therapy. We'll get some of those 40% I was talking about won't be treated, right? But commercial is what I'm told by the industry doesn't like that, okay? Because it's very expensive to develop a drug and they'd like to have the largest market possible. So you're faced between developing a drug for a wide population where it doesn't work in a bunch of the people, or having a more effective targeted therapy in a narrower population. What are you doing?
How am I solving that one? I'll just give you back to the example on the DMMR endometrial population. Sure, it's 17 to 20% of the entire population, but guess what? That therapy is durable for those individuals. Yeah, the Kaplan-Meier curve's flat again. So my argument with Luke is always duration of therapy compensates. Now, obviously, there is the other convenient piece about, and if I can look at underlying pathobiology, the tumor agnostic label is a great concept, but it really hasn't caught on yet in terms of, I'd say, the commercial environment. But I do think there are answers to that we should be able to trade breadth of coverage for efficacy and duration, because that's what patients want, right?
So I'm being told to cut it short. So one last question.
Hi, thanks very much.
Around, so very quickly.
Yeah, I'm around until I have to go off and do my homework this evening. Sorry, I can't join you for dinner.
Thanks very much. Really interesting presentation, Claire Stewart, Horizon and LJMU. My question relates to the aging population, which you mentioned, particularly the health inequalities that we see, which are so unacceptable in a first world, 21st century. But I guess my question relates to the fact that, you talked about putting the patient first, you put the patient in the center, the need to be able to do that. You're a keen cyclist, you're CSO at GSK. What are you doing to enable those patients to actually undertake lifestyle changes? Because we fundamentally know that if you increase the amount of exercise you're doing, you alter your epigenetics, you push back the disability threshold. So rather than trying to use drugs which are actually going to lose efficacy because people in 20 or 30 years aren't going to be as healthy as they are now in their 70s and 80s, what are you doing to change the lifestyle of those individuals to be able to make sure that the drugs that you are developing are for those crucial diseases like COPD, like cancer, which aren't avoidable potentially?
Yeah, what I would say is, look, this is an ecosystem problem. What I'm set up to do is to find solutions when those behavioral changes don't work. So what I'm doing about it is focusing on the burden end of disease, the underlying, and also quite frankly, although I talked about subdivision, being realistic about the point that when it comes to treatment, there's not a sophisticated phenotype available in the surgery of a physician in the valleys in Wales. So what we're trying to do is create medicines which address burden in as broad a population as possible, remove the need to worry about that phenotyping from a label standpoint. We made our life hard, if you like, in our COPD studies for IL-5, because we included there are various groups of patients who I knew the mechanism would be less effective for. How we change the overall behavioral component of what we do is a problem that I think comes from us providing data to inform choices. So what I'm doing to help there is being willing to invest in things like our Future Health and others. We're back to the point that I made. We all want it, none of us can afford it, and we get different benefits from it.
Great. Thank you so much, Tony. Fantastic talk.